Hello and welcome to our webinar on RAYSHAPE aberration correction for deep tissue imaging. My name is Bastian Klußmann-Fricke. I'm an application specialist at Abberior Instruments, and we are here at the Johannes Kepler University in Linz to pre-record this webinar. And in the beginning I will talk a little bit about technical details of RAYSHAPE aberration correction and how it is implemented in our high-end microscopes. And after that, Dr. Maximilian Janssen will give a presentation on the science they are doing here and how they use RAYSHAPE and other modules in our microscopes to greatly enhance the image quality they get out of their samples. So in the beginning of my presentation, I talk a bit about optical aberrations, how they occur and why they are particularly relevant for STAT imaging. Then I tell you something about how one can deal with optical aberrations and some technical details, how we correct for them in our high-end STAT microscopes. In the end, I also show you some examples where we used RAYSHAPE to greatly enhance the image quality in challenging samples we got during our demos. So we all encounter spherical aberrations when we work with confocal imaging. When I had my first microscopy courses in the university, I recognized that the deeper I went into my samples, I saw a significant decrease in intensity and sharpness in my samples. We see that here in this vertical section through a plant root. We have here on top a pretty nice signal, but the deeper we go, everything gets blurry and a little bit messy. Spherical aberrations are induced by refractive index mismatches. We all know this from physics class in school, and where in this case the pen in a water glass is quite a popular example. So here the pen is in two different media, air and water. They have different refractive indices, and because of that we see the shift here within this pen. And in microscopical samples, those mismatches occur between the immersion medium, the mounting medium and the sample itself. And within biological samples, they are induced by different sample components like protein, lipids, or in some cases like cuticle or something. But what happens to our images when we have spherical aberrations in our samples? So we see this here in these fluorescent beads, which are embedded in a 3D matrix. So we have again a vertical section through a sample in the depth of the sample. And as long as we are close to the cover slip, everything is fine. We have a nice signal intensity and well-shaped PSFs here. But the deeper we go into the sample, we see a decrease in fluorescence intensity and sharpness in our PSFs. And what we also see here is if we look into deeper sample regions, is that the loss in sharpness is to a great extent because the PSF is smeared out in the Z dimension. And this is the reason why spherical aberrations are highly relevant for STAT and for 3D STAT in particular. But why is this the case? So when we consider our 2D and 3D STAT donuts, as long as we are close to the cover slip, up here for example, everything is fine. We have no aberrations. We get nice STAT donuts with a well-defined intensity minimum in the center. And this way we get the highest possible resolution, highest possible signal, and a very nice signal-to-noise ratio. We see here, this is again our bead sample, and this is a 3D STAT image of it. And here in the upper region we get nice and isotropic super resolved beads. However, we also see that as soon as we go deeper than 10 to 15 micrometers into the sample, the image gets blurry and you basically don't see anything anymore. And the main reasons for this is that also the STAT donuts are affected by the spherical aberrations, and they get more and more distorted because of the mentioned refractive index mismatches. So also the donut gets blurrier and smeared out, particularly in the Z dimension. This holds true for both STAT donuts, but it's not that tremendous for 2D STAT. For the 3D STAT donut, however, a smear of the depletion light in the Z dimension basically means that you get depletion light into your intensity minimum in the center of the donut, meaning that all the fluorophores in the focal plane are depleted. So in the end, you don't see anything anymore. But what can one do to overcome spherical aberration in samples? So one option that has been used was to apply different settings for the correction color that is available for certain objectives. And you see this here on the left. And they were originally being used to adapt for different thicknesses of cover slips. Of course, you can use this to also correct for aberrations that occur in your samples, but this unfortunately comes with a list of disadvantages. And one we see directly here in those images. These are stained nuclei in a Drosophila embryo. And by using this method, you always have to choose in which area of your sample depth you want to have the correction working. So either you have a nice image here on the top, in the middle of the embryo, or completely at the bottom of your sample. Besides that, it is only available for certain objectives. Normally, silicon and water objectives, which have a lower NA than oil objectives, for example, and therefore the resolution you can achieve is much lower. It's furthermore a static correction, meaning that you can't make any changes during a running measurement due to focal shifts you would see. You furthermore have hysteresis effects, and you have to optimize the settings basically on every single C position in your sample. However, there is also a better and cleverer way to correct for spherical aberrations in your sample, where you don't have all those disadvantages. And this is what we call RAYSHAPE, where we use adaptive optical elements to shape the wavefront of our lasers and the detected signal to correct aberrations that occur in our sample. And this works dynamically across the whole sample depth. You see this here in this image. We have a nice and clear image across the whole Drosophila embryo in this case. And to achieve this, we use deformable mirrors, which consist of a reflective membrane, which can be used to modulate the wavefront of a laser beam. Actually, the concept is adapted from astronomy here. You see here one of those larger deformable mirrors that are used in telescopes. And they are used to correct for atmospheric interferences. The deformable mirror itself consists of a reflective membrane, and below that we find a number of electrodes here. And by applying different currents, we can either push the membrane away or pull it to the electrodes. So like this. And in the end, by applying different currents to all the electrodes in the deformable mirror, you can give the mirror a specific shape, and this way you can correct for the occurring aberrations, and you can highly improve your image quality. And here we get a nice image of Uranus. So before that we have the uncorrected image, which is very blurry, and after the application of a deformable mirror, you get a nice and sharp image. And we at Aberrations adapted this technology to our high-end microscopes to give you the best possible image quality across your whole sample. But how does this work? As I already told you, in conventional imaging, the image quality decreases significantly the deeper you go into your sample. This holds true for normal confocal microscopes without in the deformable mirror, or here in this case where the deformable mirror here is turned off. And this is so the distortions and the distorted PSFs you get because we have our laser light here coming into our optical system, and we have a continuous wavefront, and it stays like this until the light enters the samples. There, because of the mentioned refractive index mismatches between the immersion, the mounting, and the sample itself, the wavefront gets aberrated, and you get a distorted PSF in your focal plane. However, the same holds true for the emission light, which also gets distorted, and in the end, you get a low signal at your detector and a much lower image quality of the acquired images. But in our aberration microscope, we can use the deformable mirror to greatly improve the resulting image quality. So we have our deformable mirror here, and we do this by dynamically changing the shape of the deformable mirror depending on the current focal plane within your sample. So while we are scanning into the sample depth, we constantly change the shape of the deformable mirror to compensate for the aberrations occurring at different sample depths. So we pre-shape the rays before they enter the sample and get an optimized PSF in all Z positions in the sample. And the deformable mirror is furthermore also able to correct for aberrations affecting the emission light that comes out of your sample and goes into your detectors, resulting in a high signal. And this way we greatly improve the image quality we get in the images we acquire with our microscopes. So here we see that in direct comparison, we have again a vertical section through a plant root. Here on the left we have the uncorrected image, and you see a severe decrease in fluorescence intensity and sharpness down here in the lower parts of the sample. But when we apply RAYSHAPE aberration correction, we get a much sharper and brighter image down here, where you can see individual cells and even their substructures. RAYSHAPE in our systems can easily be applied to your measurements. You should just set it up in the beginning of your session, and it dynamically adapts to all position and depth in your current sample. It's very fast. You can easily optimize the settings on the fly while running your system in live mode. Furthermore, it works on all laser beams, the stat lasers as well as the excitation lasers, and of course also for the emission light. And last but not least, it is super easy to optimize within our Lightbox software. You just activate the deformable mirror here, and then you have those two sliders. And you have one slider for the top of your sample and one slider for the bottom of your sample. And this way you set up a gradient for your correction, so that you have a dynamic correction across the whole sample depth. So I want to sum this up a little bit. So when we do confocal and stat imaging in particular without any correction, everything is fine as long as we are staying close to the cover slip. But as soon as we go deeper in our tissue samples, the stat energy is leaking into the intensity minimum, and this way you lose resolution, signal intensity, and get a poor signal-to-noise ratio. And this is particularly relevant for 3D stat imaging, because you basically lose the intensity minimum in your stat donut at all, resulting in the depletion of all the fluorophores in the focal plane, and you basically get close to no signal in the deeper sample areas. However, when we apply RAYSHAPE, we can minimize the effects of the occurring aberrations in our stat donuts, and therefore we maintain the resolution, the signal intensity, and get a good signal-to-noise ratio across the whole sample depth in our whole image. Like we have in this image of the 3D beads here again, we see our nice super resolved beads across the whole sample depth. And using RAYSHAPE comes with many advantages compared to other methods for aberration correction. So first of all, it works with a wide variety of objectives, meaning that you always have the best possible resolution. We have a dynamic correction across the whole sample depth, which is dynamically applied during a running measurement. As the deformable mirror is an electronic device, you also have a high reproducibility. So with a sample that I have worked before, I can even apply the same settings as before, and it more or less instantly works. So you set it up once in the beginning of your session, and it works everywhere in your sample. And finally, we have our very easy to use and straightforward Lightbox software, which very nice and easy RAYSHAPE integration. So now I want to show you some data where we used RAYSHAPE to greatly improve the image quality in challenging samples we got during some of our demos. So here, for example, we have a vibratome section of a brain slice. This is just a confocal overview, but then I acquired another images. We see that here, so this is the position where the image is taken, which is again a vertical section through our sample. So and here, if we don't have any correction, we get for confocal decent image on top, but it gets blurrier and blurrier and blurrier the deeper we go. However, if we apply RAYSHAPE aberration correction, we get a much nicer and sharper image. And this, of course, holds also true for 3D stat. Here only for one color, the piccolo in this case. Without aberration correction, we basically only get a nice signal up here in the top part and lose the signal more or less completely in lower parts. With RAYSHAPE aberration correction, however, we get a dynamic correction across the whole sample depth and get a nice and super resolved synapses in this case, even in the greater sample depth. So the next example we have here is embryo of the nematode C. elegans. They are actually notoriously hard to image because they have this cuticle here. So is a cuticle that is located around the embryo, and this has some really weird optical properties. So for confocal, you get a decent image, but with 3D-stat you basically don't get anything at all. But if we apply RAYSHAPE, we get those images. So even confocal looks much better, and we get a really, really nice 3D-stat image here across the whole sample depth. Here we have that in direct comparison, so 3D-stat without any correction, you basically don't see anything. But with RAYSHAPE aberration correction, we get a nice staining of our nuclei here. And then, when you have set up all the settings perfectly, then you can even acquire those nice little videos here. So we have a nice super resolved nuclei here with the nuclear pores and the lamine around the nucleus. And RAYSHAPE aberration correction can also greatly help for live cell imaging. So we had one demo where we unfortunately didn't have a water objective available. So meaning, as I mentioned before, as long as we are very close to the cover slab, so we have living cells here, as long as we are very close to the cover slip, everything looks nice, especially in 3D-stat, but as soon as we just go a few micrometer in our aqueous cell culture medium here, everything gets distorted and you don't see much anymore. However, if we apply RAYSHAPE, we get a nice 3D-stat image across the whole cell, and we can even compensate for the very high refractive index mismatches between the immersion oil and the water in the cell culture medium. So here we have that in direct comparison again for two colors in this case. We have here mitochondria and DNA, and without correction, again, close to the cover slip, everything is fine, but as soon as we go a few micrometers into the sample, nothing, you can see more or less nothing anymore. But with our RAYSHAPE aberration correction, we get a nice 3D-stat image across the whole cell. Okay, now I want to sum this up a little bit. One of the great advantages of our microscopes is that basically all the different modules we offer to optimize your images are combinable. So here, as I said, as a summary, if we use confocal without adaptive optics, the deeper we go, everything gets blurrier and messy. But by using adaptive optics or RAYSHAPE aberration correction, we get a nice and clear image across the whole sample depth. Also, I said that before, 3D-stat without adaptive optics, after just a few tenths of micrometers, you don't get an image at all anymore without aberration correction. However, if we use our RAYSHAPE, we get a nice and clear image across the whole sample depth. And on top of that, we also have our matrix array detector, which we use to reduce occurring background in our samples. And we can combine those two to great effect. We see that here. We have, again, 3D-stat with RAYSHAPE aberration correction and the matrix detector. And this way we can even reduce the confocal background we have in our samples and get our nice and isotropic super resolved fluorescence beads here in the deepest sample area, which are in this case roughly 100 micrometers. And with this, we want to come to the second part of our webinar on RAYSHAPE aberration correction. I welcome Dr. Maximilian Janssen from the Johannes Kepler University in Linz. He is a research physician here, working part-time in science and part-time as an actual medical doctor. And he now wants to give a talk on the research they're doing here. First, I want to ask you some questions about yourself. So, how did you come into contact with microscopy and super resolution microscopy and science in general?
Well, good question. I was studying research basically when I was doing my medical thesis in Germany. I was studying at Heidelberg University, and my former boss there, Maren Engelhardt, is a neuroscientist who works a lot with confocal microscopy and with nanoscale imaging. And by studying my medical thesis there, I also started to learn more about confocal microscopy. And when she got the job offer here to be a professor in Austria, she offered me to come with her and to kind of build up the lab and build up our stat system that we have here. And yeah, that's how I came to my position.
Oh, very nice. And also, so a research physician, how does it actually work? So you're a medical doctor working in the clinic as well as a researcher. So first, how does it work at all? And how can you cope with the workload? I mean, being a doctor is quite quite a bit of work and a researcher as well. So how can you cope with this?
My job model is kind of a split time concept where on Monday and Tuesday I'm in the clinic. I work as a resident in the ENT department over here. And from Wednesday till Friday and on the weekends I am a researcher here at the Institute. And yeah, how it works it depends a lot on my team, and we need a lot of support, of course, to make this possible. The clinic needs to be accommodating to my times that I have. And I have my whole team over here that's supporting me in the days when I'm not here, and to kind of do the work in the times where I cannot be present in the lab. But in the end, with a lot of organization and structure, we get it done most of the times.
Yeah, very nice. Okay, so now you will give a presentation and show us some nice images where you use RAYSHAPE aberration correction to greatly enhance the image quality you get out of your samples. And you will, of course, also elaborate a little bit on your science you're doing here. Thank you very much. And it's up to you.
Hello again, and thank you to Abberior for coming over here today to film this webinar with us in JKU Linz, Austria. I want to start today by briefly talking about the backstory of our research and why we use nanoscale imaging to answer certain questions about neuronal behavior. Starting to the topic, I want to briefly talk about neuronal activity and synchronous behavior. Right now, all of you are probably somewhere on the scale between sleepy and arousal, hopefully more on the right-hand side of the scale. And your brain is cycling through different states of activity and of synchronous behavior all throughout the day. And it takes a huge amount of good balance and of neuronal interaction to keep this balance alive and to not slide into any side of desynchronous behavior. If we have enhanced inhibition inside the brain and we lose synchronous firing, we can see certain pathologies slide down into coma and lastly into death. And when we have heightened synchronous behavior within the brain, so we see a lot of neurons firing together in a constant feedback loop, we see disinhibition and overactivity in the brain leading to seizures and epilepsy, and in the end, oftentimes death as well. And there are several theories and also several models of why epilepsy can develop within the brain. Some of them are due to gross changes in the neuroanatomy of certain neuronal areas, and some are related to small aberrations or genetic changes in single channels or in single connective areas of the neurons themselves. When we look at the electrical field potential, so on the electrical activity that we can record directly from the outside of the brain, what we see is these synchronous brain waves that most of you probably know as the beta waves, gamma waves, and so on. So the neuronal firing patterns that change depending on your state of attention, if you're in deep sleep, in REM sleep, or in heightened focus on something. What we oftentimes don't think about is how these field potentials come into place, because for every local field potential to develop, we need thousands, if not millions of neurons firing at the same time and communicating with each other to behave in a synchronous way with each other. And most of these brain waves, so the ones we're talking about here, the gamma waves, beta waves, are quite well understood in the way they come into place and in the way the neurons interact in order to reach this synchronous behavior. But there are other types of brain waves, these so-called high-frequency oscillations or ultra-high-frequency oscillations, where it is not yet fully understood how they can develop and how brain cells over large distances in the brain, so several centimeters apart, can synchronize themselves in a manner that they fire almost instantaneously. And it is believed that there are certain ways of neuronal communication besides the normal chemical synapse interaction that make these interactions possible. The reason I got in touch with this topic is that the lab where I started my research in Germany is focusing on alternative ways of neuronal plasticity and of neuronal communication. We do a lot of our work on the topic of the axon initial segment, so a specialized nanodomain that's located within the first few micrometers of the axon after the soma. And it is a specialized structural domain that consists of a huge amount of ion channels that are focused in this area. And as we can see over here, this is also the site where specialized GABA-erg synapses between our pyramidal neurons and inhibitory interneurons, the so-called chandelier interneurons, can form alongside the AIS. We can see in this reconstruction of a normal confocal image our AIS in gray and the GABA-erg synapses forming between GABA-A receptor alpha-2 subunit and VGAT in blue and in red. And we also have some of our early confocal and 2D STAT images showing these GABA-erg synapses alongside the AIS. And via these contact sites, our inhibitory interneurons get a huge influential input on the pyramidal neurons and can, in fact, decide on their activation or inhibition, so if our potential will fire or not. And all of these specialized substructures make the AIS very interesting for us and for other research groups in investigating new ways of neuronal communication and of synchronous behavior. There has been some early work in the 90s already about neuronal ensembles and the influence that direct electrical coupling could have on high-frequency oscillations within the brain. And as we can see in these earlier decoupling experiments, there are some cells within the hippocampus that seem to have direct connection along their proximal axons that allows them to almost instantaneously synchronize with each other and show these directly coupled behavior and this synchronous activity without the need for a chemical synapse to transport the information from one cell to the other. So this makes it more and more interesting for us to see how specialized nanodomains within the proximal part of the axon can be relevant for neuronal communication and for the synchronous behavior of axons that are in close proximity to each other, or in close proximity to other cells within the neocortex that could be able to facilitate synchronous activity in between the neurons. During my early days in the lab, I was focusing my work on an even less understood nanodomain within the axon and within the AIS that is called the cisternal organelle. The structure, which you can see over here in green in the upper left picture and in magenta in the STAT image down below, is basically a stack of smooth ER that is always found within the AIS and usually in the ankyrin G deficient sites, so in the parts of the AIS where our structural proteins show some gaps in their cytoarchitecture. And there have been a few research papers already looking into structures that co-localize with these cisternal organelles and trying to figure out what their function could be for the neuronal connectivity or for the homeostasis of the AIS. And while most of these structures are known to co-localize grossly with the cisternal organelle, it is not yet proven that they actually form contact sites with them or in what way they allow for interconnection between the AIS and any synapse that might dock onto the structure. And in order to get a better understanding of this, we now started to employ several ways of nanoscale imaging. When I was starting out in Germany with this project, we didn't have a STAT in the beginning, so we had to find other ways to allow for nanoscale imaging. And what I was starting out with was expansion microscopy approach, which means that basically instead of finding a way to zoom in more into our structures, we would instead expand our sample by ×4. We will immerse our brain slice that we have into a monomer solution and then polymerize this solution in a way that allows us to bind all proteins and antibodies on a nanoscale level, and afterwards, by just adding water to the gel, we can tear all structures apart from each other by ×4 and thereby allow 4 times higher resolution of our structures in the end. Even though this is a quite laborious process that took several months to actually get going and always takes 1 week to allow for one single image. We got quite nice images with that, and we were able to localize our cysternal organelles within the gaps of anchoring G at the AIS. And we also got quite nice 2D and 3D images of that. We still had the limitation that with this workflow, we could only get a very low throughput of our data. And in the end, all antibodies that were quite low binding, that didn't have the brightest signal, would get lost in the process. And by tearing the structures apart, we would lose all antibodies that have lower signal intensity. After moving to Austria, we acquired a facility line from abberior here at the JKU. And we are now able to take our imaging approach to a new level, and actually also moved away from our rat and mouse models that we had into human samples that we get in a cooperation project with the Neuromed campus over here. So when there is tissue from neurosurgery that has to be taken out by means to get to a tumor or to get to an aneurysm within the brain, we will now use these parts of brain that would have been thrown away before and use them to study healthy neuronal structures and to confirm if what we see in mouse models and in cell culture actually also works the same in human brains. And what you can see over here is quite a section of a human neocortex slice that we imaged here in Linz. And by zooming in, we can nicely image the single neurons. We can see the AIS here in cyan blue and our cysternal organelles alongside the AIS in magenta. And now this is what we would previously get as normal confocal resolution. Now applying 2D stat microscopy, we can actually see this lattice-like structure of our structural proteins, and we can see the small parts of the cysternal organelles that are clustering in these deficient structural sites in between. So again, normal image quality below and stat image above. And the huge benefit we have from this is now that our workflows are way shorter than they used to be before, and we have a much higher throughput, and they're actually able to image one AIS in 1 minute instead of 1 hour, as it was with the expansion microscopy workflow. We now also employed 3D stat workflows on these samples to get a better understanding of the spatial location of ion channels and of certain synaptic proteins along the AIS, and to prove and see if they colocalize along the outer membrane of the AIS or if they actually go deeper inside the core of the external structures. In order to do so, we now combine high-resolution 2D stat images with axial imaging. That means instead of imaging whole 3D volumetric data, we take 2D stat image of our neurons and of our AIS structures. And alongside these yellow marks you can see in the lower picture, we are now able to take an axial cut through our axon and show and prove whether certain structures like the KV21 channels are actually clustering within the deficient gaps of the anchoring G or beta phosphocespectrine, or if they lay below or above, and how close they get to our cysternal organelles. Because as you can see in the upper picture on the right hand side, the cysternal organelles tend to oftentimes be quite central inside the axonal structure, but they are always moved way closer to the outer membrane whenever there's a contact site with ion channels or with other structural proteins that are on the outside of the AIS. Going back to our previous model and to our question, what modulates network states, we also look at different pathways and different key players within the brain that could play a role in regulating neuronal activity and synchronized behavior. One of these cells we look at are microglia, which are known from previous experiments of other groups to be highly regulated in, for example, anesthesia and also in epileptic models of epilepsy, where we see, in both cases, heightened activity of microglia and actually large activation that leads to them modulating the neurons around them. And one interaction we look at is, of course, again, the interaction of microglia with our axonal segments. And it could be shown that there are preferential contacts of microglia alongside the AIS, and by taking optimized confocal images and 3D segmentation, we can measure and quantify these interaction sites and take a look at these sites if they change in their size or in their frequency in models of heightened excitation, like, for example, epilepsy or after spinal cord injury, where our cells within the brain are still intact even though the axon way far down in the spinal cord has been cut. Now, after optimizing imaging for confocal, we also want to take a step further and see if we can resolve the nanoscale structure of these microglia AIS interaction and to see if they actually prefer these AIS or these structural protein deficient sites or rather cluster kind of randomly alongside the AIS. And we combine these images with several key players that are known to form the outer membrane and the kind of protective structure of our neurons and interneurons, like the perineural nets that form large proteoglycan networks around the neuronal body and the dendrites and the axon, and thereby work as kind of a protection layer that keeps microglia from attaching to the AIS or to the neuron body and keeps it from modulating any synapses that could be structured alongside the AIS. Finishing up, to not take too much of your time, I want to briefly show some examples of RAYSHAPE, matrix, and other modules we use on our SLAT microscope to optimize confocal images and also SLAT images. Because most images you saw right now and most SLAT images, we always employ the modules that we see fit, and it was kind of hard to find before and after of what can be done with the SLAT microscope. And over here you have a nice example of 3 raw images that I exported directly from the SLAT without any post-processing. And you can see on the left-hand side the big challenge we have with our human samples, because all of our tissue that we get is kind of thick. We have a 300 micrometer vibratome slice in this example, and the deeper you go inside your sample, the more you will use contrast and lose photon density and intensity in the sample. And 2 modules we use frequently to kind of compensate for that and to get a sharper image is the RAYSHAPE module. So by adapting our deformable mirror in the beginning of the imaging, we can have it move alongside the Z layer. So as we move deeper, it will give us more and more aberration correction, the more our sample gets out of focus in the process. And lastly, if we want to get the last bit of intensity and contrast of the sample, we will employ the matrix as well to capture all light that's in focus and out of focus. And thereby, as you can see, if you look at the small filaments of the cells, you have heightened contrast and you will have more sharpness in the back of the image that is more down in the Z layer of our sample. Maybe one interesting feature that people will also need if working with human samples is the time-bow imaging, because we had huge challenges with our old human tissue, since neurons within the brain will stay alive for years and years and years and thereby accumulate more and more debris within them, which we see in our images as lipofuscin granules. They are the large granular structures here in the center, and usually they produce background in most channels, like in the green and in the orange to red channels, and it's quite tricky to work with them and to get them out of our sample. What we did to get a clear image and to cut out our background signal is to deploy time-bow on our matrix detector and filter out all photons that came with a long fluorescent lifetime or with a short photon arrival time, and thereby segment our antibody signal from the autofluorescence signal of the lipofuscin granules. And as you can see in the next slide here, this works quite well on all channels. We have a 488, 594 and a 647 antibody in this sample, and the lipofuscin signal could be filtered out of all of them, and we get a clear image that is much closer to the optimized images we get of our perfused mice or rat samples from before. With that, I want to conclude my little presentation. I want to thank, again, my whole team here at JKU Linz that makes my work possible and makes these pictures possible in the end. And yeah, if you have any questions afterwards, I'm looking forward to answer them. Thank you.
Welcome again. Now for Q&A. So first things first, please write down your questions in the corresponding tab, and we will try to answer all of them. Although if we are not able to answer all of them, you will get a written answer in the next couple of days. Hi, Max. Nice to see you again. I think I will start with just two short questions. So how does it work in Austria? So do I get it right that you can basically use all biopsy samples or waste products of surgeries for scientific use? Is that right?
It depends. You still have to do an ethics vote and all of that to work with the samples. But compared to Germany, in Austria, everybody is a body donor if they don't say otherwise for samples. So whenever there is tissue that otherwise would be discarded, that's not needed for diagnostics for the patient, and that would have a use in research or in sampling, then after receiving an ethics degree, we can use the sample and anonymize them for human research.
Mm-hmm. Okay, that's more or less also to some extent the dream of every researcher working in a biomedical context, right? To just get your human samples out of the Another short question with the images you showed with matrix and RAYSHAPE. Do I get that right, that these have been acquired volumes and then you made a maximum intensity projection? Or
yes, I didn't show the volume. I just showed the MaxIP from the front of them. Mm-hmm. So this was like a thick tissue sample going inside, and then I just added the background to see like what appearing more and less when you activate the RAYSHAPE.
Mm-hmm. Okay. Nice, thank you very much. So then I think I start with the questions we got from our visitors. So one question we got already during the talks is the question if also the excitation lasers and the depletion lasers are affected by the deformable mirror in the same way. And this is, yes, this is true. So the deformable mirror works basically on all the excitation beams, so as well as the depletion beam. It furthermore also works on the emission light, so it also corrects for aberrations that affect the emission light, and also corrects them to get the best possible image. Okay, so and then the next one was the question if there is a difference between the Lightbox and the Inspector software. Yeah, this is true. So our Lightbox software is easy to use, straightforward imaging software that normally runs in the foreground of our microscopes and is suitable for, I would say, 99% of all the imaging and most of the users. And there we optimize the settings for aberration correction, the RAYSHAPE, to be really straightforward and easy. So you saw that during my talk. You have those two sliders, you activate the deformable mirror, and then you have basically in the beginning of a session, you make an XZ section through your whole sample, and then you set correction values for the top and the bottom of your sample, and you can reuse all those settings over and over again across your whole imaging session. And this works really well. I think I mentioned that you always, even if when you have an older sample you already worked with, you can reuse the setting you used before, and this normally works fine, as long as you don't have an air bubble or something like that in there. However, then we, of course, in the background runs our expert software, which is Inspector. We didn't cover that during the webinar here. Which is by far more complex, and you can there really have access to all the different functions of the deformable mirror. You can also, with Inspector, you can also improve, for example, the adaptive optics even a little bit more by using our SLMs, which we use to generate the stat donuts. You can even improve the quality of the correction even a little bit more if necessary, although we figure out that in most cases the deformable mirror is completely enough. But this, I would say for most users, the inspector software is far too overwhelming, because in most cases there are biologists sitting in front of our at the systems, and even so I'm a biologist myself, so the inspector can be a little bit overwhelming, and you have to know what you're doing. But you can, if you have one of our systems, you can get a special training for that and also use that one if you like. Okay, I hope that answers the question. Then let's see what we also have more questions here. So could you discuss any limitations or trade-offs associated with using deformable mirrors for aberration correction in stat microscopy? For instance, are there any compromises in terms of imaging speed or spatial resolution? I think to answer this question, this is the beauty of using a deformable mirror. So there are no compromises in terms of imaging speed. So as I said, the deformable mirror is an electronic device, and it can change its shape very, very fast. So it's really for each individual Z position during even an XZ scan, it can change its shape for each individual plane that is acquired. So it's really, really fast, and you don't lose any imaging speed, and of course also no spatial resolution normally. Then here is a little bit of off-topic question. Give me 1 second. What specific types of aberrations are most effectively corrected by the deformable mirror in thick samples? Okay. It is to the topic. Sorry, I misread the question. So we normally figure that we only face spherical aberrations of the 3rd order in biological sample, in even more complex biological samples. So, and this is also what is correct. What the sliders for the setup in the Lightbox software control. But within the inspector software, you offer, and underlying there are Zernike polynomials to have the bias aberrations that are controlled. However, as I said before, in inspector you can use more higher order. You can correct for higher order aberrations, but we figured out that in most biological samples, they don't play a tremendous role. So we have considered putting even more controls in the Lightbox software, but we didn't find that was really useful, and just makes the process more complicated. Okay, then to the next question. Can we export the image? So I take an easy one. Can we export the images in XZ or YZ position? And if yes, then how? So this is easy. Of course, we can do an export of, so first we can acquire images in that way. So we can just make one single scan in one section. So these more or less all the images I showed during my talk were XZ sections, and you can just acquire them in that way. And then of course just export them as any other image. And furthermore, of course, you can, if you acquire a volume, you can just export the whole volume. And if you do 3D stat, you have an isotropic resolution, and then can later on export individual images by reslicing an image tray, for example. So that's, of course, also be possible. Okay. So then the next question would be, can you please explain a bit more of what ray shaping is and how it works? Is it similar to detector side adaptive optics, which involve a form of a waveform detector and a deformable mirror? So no, we are doing sensorless wavefront shaping of our lasers. So the downside of it would, of course, it would be possible to do a wavefront sensor into the microscope, but this would come with the first the expense of imaging speed, because you need to process the data you get from the wavefront sensor and then have to feed it to the deformable mirror. And furthermore, you also would have an additional beam path in the microscope to where you get some of the light and feed it to the wavefront sensor. And this way, you lose basically signal intensity. So we also didn't saw the actual benefit of putting a wavefront sensor into the system, because this would make the process even by far more complicated, and we wanted to have it as easy as possible and straightforward. Yeah, and this is why we didn't do that. Further questions? Okay, here is a question for suggested dye pairs for time bow imaging, which is a little bit off topic. The one thing we can recommend is our Star Orange dye and CF594 by Biotium, just as a recommendation. There are others out there. We don't have a specific chart for that. There you unfortunately have to do a little bit of your own research. Yeah. Yeah. How do you decide the best shape of the deformable mirror without any guide stars in the tissue? And this is, as I said before, it's an iterative process. Normally, you go for make an XZ section in the beginning of the session, and then you optimize for signal intensity and sharpness by eye. You normally do an iterative process for the top and the bottom of the sample, and then you can reuse those settings over and over again. Do you have any comments on that, Max? How do you do that?
Yeah, but the good thing is there, I also usually start, as you said, iterative and take like a huge sample and look for the best focus. And afterwards, when I do several samples in different Z layers, the mirror automatically adjusts to wherever you go. So for example, I take 3 images in 3 different Z layers. It will always go back to the first measurement you took and where you optimize, and then it will just take the mirror range from this bigger image you take. So you can take some time at the beginning, adjust, and afterwards we're just going to go the sample that you have.
Yeah, as I said before. So you basically define the top and the bottom in the beginning, and then you can, it will automatically, this is also why it's called adaptive, right? It adapts to each sample position and each sample depth. Here we have another question for you. Here it states, a stupid question from a non-specialist. How many exemplars of brain cells commonly one can get from one human for the research? Thank you.
Yeah, good question. And it depends really on what surgery you have, of course. In some cases, if we receive samples from epilepsy surgery, for example, the surgeons have to remove 2/3 of the temporal lobe of the patient on the side. So you can imagine getting like a chunk of 3 to 5 centimeters of brain that you can work with, which of course, the patient is still fine afterwards. Of course, they have some minor impairments, but way less than what you would expect from taking out so much of the brain. So the regular thing we get when we have excess tissue from surgery would be like maybe 5 millimeters cube of sample, which still leaves you with, depending what sectioning you do, 20, 30 sections with thousands and thousands of neurons inside.
Yeah, okay, very nice. As I said, it's nice to get so much human samples, actually.
Yeah, it's definitely unfortunate to do it.
Yeah. Next question is the deformable mirror regulated for each imaging channel separately during the multi-channel imaging? No, it's optimized basically for you. Although the spherical aberrations are to a minimal extent dependent on wavelengths of the excitation light, we normally, if we have a 3-color, we use the wavelengths in the middle, and you see only minimal differences between the individual wavelengths, and you normally use the one in the middle and get pretty good results for all the wavelengths. Then the next question is the aberration correction automated, or is it dependent on the user to determine when a good correction is applied? And this is what I meant before. This is up to the user, and a lot of those questions we got here are in that direction. So first, we do that by eye. I said it, as I said before, it's an iterative process. You take several images with certain settings for the top and the bottom, and then you basically use the best one. And then, of course, here's the question: that if there is a certain metric resolution or intensity in the image? No, I mean, we are specialized in stat imaging. And if you try 3D stat, for example, as soon as you're in deeper sample areas, if your deformable mirror is not properly set up, you don't see anything. You don't get a signal, and you don't have you basically don't have a signal. So for deeper sample areas, you have to have good settings to get a good signal out of your sample. So this is actually a pretty good metric, I would say. If you're able to do a 3D stat image in a deeper sample area, then your deformable mirror is probably set up. And then there's one more question. Is the wavefront correction based on a Zernike polynomial? Yeah, these are the bias aberrations that are underlying in the deformable mirror. These are Zernike polynomials. So, yes. Thank you very much for your attention. I hope we could show you the beauty of ray-shape aberration correction. And I said that already several times. It's really easy to use. You set this up in the beginning of your session, and it works all the time. Yeah, again, thank you for your attention, and have a nice afternoon, as it is here in Germany, or a nice start in the day in the US, for example. Okay. Bye-bye.
Bye.